Good morning, everyone. On behalf of Jeevasa Healthcare USA, I welcome you all to the first international workshop on stem cell science and regenerative medicine, organized in collaboration with the Punjab University. It's my pleasure to introduce Dr. Sunirmal Sheet, research professor at the National Institute of Animal Science, South Korea. Today, he is going to speak about the importance of bioinformatics in stem cells research. So, I welcome Dr. Sunirmal Sheet to present his work.
Okay, first of all, thank you, Dr. Shivakumar, scientist in Jiva Hospital, for warm welcome and introduction. So, good morning, distinguished organizer and respected faculty and dear colleagues, for organizing such a scientific event, first time, on International Stem Cell and Regenerative Medicine Workshop at Jiva Hospital. So first of all, before presentation, I would like to give some st kind of like letter of appreciation. So first of all, I would like to thanks Dr. Shima Rai, organizing secretary, Punjab University, then Joydeep sir, uh, director of Jiva Hospital, and Mr. Deven Patel, founder of Jiva Hospital. So, and all over, I would like to thank the organizing committee for their exceptional efforts and support. So, and with that, special thanks to Dr. Shivakumar for the kind support, guidance, and coordination. So, with appreciation, Dr. Sunirmal, I am a assistant research professor in National Institute of Animal Science, South Korea. So, basically, here I am going to present overall the importance of bioinformatics in modern as a modern technology in stem cell research. So here is my topic, like importance of bioinformatics in stem cell research. So first of all, as we know that with research progress, the modern technologies, especially in the bioinformatics, in case of genomics, the techniques also slowly uh, improved. So in that case, so first of all, like why the stem cell and bioinformatics, why it is synergy? We know that especially the stem cells, it is kind of heterogeneous. So in case of that, we need high throughput platforms such as bulk RNA sequencing, single cell sequencing, epigenomic profiling, and advanced imaging through bioinformatics tool for the complex stem cell datasets. So therefore, why we need is because bioinformatics generally it provides quantitative, unbiased method for analyzing cellular heterogeneity and reconstruct the lineage decisions, the fate of the cell and the lineage from where it came. We can diagnose or we can identify that. So based on that, we can use further for therapeutic purpose. Next, I have given this is like kind of overall bioinformatics framework for which developed in the recent year will help for the analysis and promote the application in terms of stem cell medical research. So here we can see that multiple techniques or multiple approach in terms of genomics, transcriptomics, proteomics, metabolomics, and epigenomics. So all together, like we can approach as a multi-omics for application purpose in the stem cell medical research. So here one example. So here is like iPSC, the stem cell. Here both are mixed. For example, the male and female candidate. From there, it's a mixed population. After differentiation, the chimera organoids is forming. So here, for study purpose and for getting the accuracy of data and the population of the cell, we can differentiate the two different kind of data set. One is RNA sequencing for diagnose or for getting the information of transcriptomic changes or the transcriptomic insights. Second one is we can do the single RNA sequencing. Here we can get the uh, single cell population number and single cell changes inside the molecule at the molecular level. So there are many other things, for example, like SNP or any kind of mutation, everything we can come to know after the sequencing analysis. Next, here I have summarized some of the major tools which is useful for the stem cell research and their key features, like what about and their application. So here is first of all is CellNet, and I have given the link also where the students or researcher can go and they can get the access. These all our tools are like free tools, so anyone can access without any kind of subscription or anything. So for example, first is CellNet. So this one is like application generally. It uses the cell engineering fidelity, refine the transcriptomic factors, prediction for the cell engineering. Second is key genes. This is also another kind of tools where we can monitor and evaluate the efficacy of the cell differentiation. Second is scorecard. That is also another kind of tool where any kind of like stem cell lines intend to differentiate, that also we can figure out after the genomic analysis. Then Mogrify, that is also predict the important transcription factor for the direct conversion. EGEC, that is also cell engineering fidelity, we can access or you can get the information. Therefore, like another is like Express Essence. This is basically it's a kind of cytoscope is one kind of free software where the add-in option is there. That is, we can use the Express Essence where we can identify the critical genes for the cell type conversion. Then stem checkers, here we can information get like stemness of the user specified gene list. We can give the gene list of certain gene list and we can get user specified gene list we can give and we can get stemness information. Next is another kind of major kind of like based on the human stem cell research. I have summarized the databases. First of all is stem formatics, where we can identify the patterns of some set of genes sample within the data set present. Next is stem base of stem cell database, Hema Explorer, and blood spot. All these tools work for the human species as well as mice species, rat species, even macaw also. So here, what data generally we have to prepare for the analysis. First of all is like generally the gene expression data. Either it can be RNA-seq data or single cell analysis data or microarray data. These three data mostly people use for the stem cell research or in terms of these tools for using for analysis purpose. So here the major analytic tool or major technique which generally researcher nowadays frequently use, that is bulk RNA sequencing for finding out the transcriptomic level mechanisms. So here I have summarized the overall pipeline for analysis. Here, first of all, first one is the model organism, reference-based analysis. Second one is non-model, de novo-based expression, de novo-based analysis. So here, the Callisto or like Salmon, this is kind of without like some person does not know much about Linux or any other programming, they can use the Salmon or Callisto based. It's online also available tool. So there also they can just give the input of first raw sequencing data, which we will get from the company. Then after that, we can follow the pipeline, quality control, then trimming the adapter sequence of the which present in the genome sequence. After that, we can mapping and quantification, mapping to the reference genome we can do. Or else if someone know the kind of they have Linux expertise or some programming, then they can continue with the other softwares. For example, FASTA QC, Genomics software, HiSat, then feature counts, where we will do the countification. What? How many count reads data are present of each gene in our based on our study or based on our design. Like for example, it's simple control and test sample in terms of like control stem cell and some kind of test or any kind of disease related. So we can comparison purpose we can get, and we can get the count read data, and mostly it will match with the reference genome, the exon portion. Intron also we can do, but exon generally people use for the normal purpose. Then after that, further following study we can do with the DE-seq. DE-seq 2 is kind of normalized, and for getting the normalized data, after that we can get the genes log-fold to change, or further PCA analysis or functional study, which we can use after DE-seq or Azure, or there are another kind of tool, Lima, if you want more strict uh, kind of criteria for getting like really good genes information, then we can use Lima. So therefore, we can use these softwares. And another one is de novo, the similar kind of pipeline, but little change. For de novo assembly, we generally use Trinity and CD hit. And after that, finally, when we get the count data, we can again follow up this Callisto. Then further we can process. This I have given the link, the blue color, where anyone, any student can learn the RNA-seq analysis without any kind of, prior experience or something, and with all free. Next is like the why we typical the analysis flow, what I have already explained, and like what kind of data generally we create for our presentation for in the paper. So basically we create first, we do after QC we can do the alignment, quantification, normalization. After that the functional study we can do like cake pathway study and cell state, or any kind of attributions we can get. So these are all kind of small example of how the RNA-seq analysis and it create data or generate the data. We saw the PCA. PCA, we know that it is used some kind of statistics, so I don't want to go details like so this is like kind of we use PCA1 and PCA2. Then here it shows the clusterization of the your sample. For example, one is your test sample, two is your control, or three is another kind of test. So we can see the clear differentiation or clear separation based on their input gene expression level. Next is volcano plot. It signifies that how many genes expressed as in terms of upregulated and in terms of downregulated, and we can give some cutoff value like for like which log fold. Generally we know that gene expression we count as a fold change. So if we want that, okay, more than two log fold change is important for us, then we can set the cutoff and we can express those data or those kind of plot in the paper manuscript. So after that, next is like it is also heat map overall, how many genes has expressed in your control sample and your test sample. We can overall view, we can see. And pathway analysis, your targeted genes are enriched in which pathway, and those pathway are really like expressed well, I mean upregulated or downregulated. That also information we can get. So overall, this all information can give us, based on your study design and based on this data, we can predict the disease progression or disease mechanism. Either it can be stem cell research or any kind of biological research, it can be useful. So next is like single cell RNA sequencing, which is the latest uh, modern technologies, generally all about world, people are using with RNA-seq data, bulk RNA-seq data, people are now advanced level, they are using single cell sequencing data. So this one is more major important data for the stem cell research, because we know that for stem cell, generally it is kind of heterogeneity. So here we can get subpopulation, rare cell types, and describe their developmental trajectory, how it is in progressing. That also we can get the information. So this is I have given generally like how people analyze the single cell data. This is kind of like overall pipeline. So Cell Ranger is like free tool which can be used in Linux. So first, all our input we can give as a FASTQ files, then cell barcode extraction. All this we don't have to do. The company people, they will provide the FASTQ files. Then we have to do the barcode extraction, EOMI extraction. Then we have to match the reference alignment with the reference genome. Then gene assignment. After getting the gene assignment, we can get the clustering or the subpopulation or expressed markers. We can do that. So here mostly like in figure I have shown here in the left side that mainly what kind of main analysis is cell level, clustering, classification, and cell trajectory assignment. And right side we can see the other kind of data, that is gene level and differentiation expression inside the each population, and that gene set analysis also, like those gene which pathway it's involved, and the which pathway is progressing during the stem cell therapy or something. We can analyze that and we can get the information. So here is one of example I presented from my data, our paper, that this is overall total population of the stem cell. This is, although it's not a stem cell, this one, this is like PBMC, blood cell. So here the each number signifies the each cell population. For example, 1 is monocyte, 2 is macrophage cell, 3 is a dendritic cell. Like that we can get all gene information, all cells information. So once we get this information, further we can proceed inside that each cell what kind of gene set has changed. So this gives us like transcriptomic level and single cell level information. So next is generally we use RNA-seq data, single cell data. We sometimes researcher use even epigenomics data or microRNA sequencing data. I did not present here all the things. So the major part I have covered here. So they use machine learning for the multi-omics integration of RNA, bulk RNA-seq data and single cell sequencing data. So here, generally the common machine learning task in bioinformatics people use for the phenotypic prediction, as we know, and representation learning for from the omics data. Then multi-omics integration. Generally, we give the trained data model in terms of matrix data or any kind of faster data also we can give as input. Then we do some validation, then test out, we get the result. So similarly here, this is kind of like overall like a pipeline I have shown and overall how it works inside. So here like input file. So after the A is input file, then we do the software management. Then we do like branching. Branching means like each data we make a kind of line of. Then after line of we do the ordering. Then we follow the standard step different based on the different algorithm of the machine learning. So after that we get the accuracy level, how much ever accuracy level, when it gets better, the result also shows that better improvement. So for that we need to like sometimes how to say, like we have to optimize the condition to improve the percentage of the accuracy level. So here I have provided the tool like where people can use the NGS data through for analyzing or integration for the in through the machine learning. So next, the basic thing is like all researcher for either in terms of stem cell research or any kind of biological research, I think that people should learn the R language, which is basic. So for any kind of statistics data, for analyzing any kind of statistics or any kind of bioinformatics, the following steps, functional study or anything. For NGS, the major part is R Studio. So here, this is, I have just took a screenshot from my interface. This is kind of RStudio interface. This I am telling for the students, like which they can learn during their masters or PhD students. So here basically is like code editor. This code editor here we can paste the script which is available on the Google. We can get, we don't have too much, know that, okay, we have to learn some coding. No need, all our codes are available on the Google. We can get, we have to just edit based on our requirement, our input data in terms of the file name, where the file is located, those things. So then we can put here the code editor and this quadrant called R console. Here only the program will run and it will show the that error or anything. It will show here. This is workspace. The generated file will store here, this side, and the right side, the below corner is plot and files. Here you can see the generated files which will help you for the correction or for the further step. So here this link where people can learn easily, like her our cheat codes, I have provided here the link where people can learn all the resources for the learning our language. People can get information. Any students can visit this link or can contact me. I can give you this link so you can learn by yourself. So here the summary of like bioinformatics studies which they have used like stem cell for differentiation of iPSC cell and ESC pluripotency stem cells. So basically the gene expression profiling, it shows that kind of distinguishing between two different kind of, this is I have given just for example, two different kind of stem cells. It was like result was inconsistent. So functional assay was not much useful. So whereas epigenomic profiling also, it shows that inconsistent result. What is mean inconsistent means, if metadata, if 10 study has published kind of gene expression data, the result found that one or two or three, there is no kind of consistency. Some gene or some pathways, one study shows that in terms of some gene, some disease, it has gone up. Some study shows for same disease, it has gone down. There, that based on that, I have like people has reported that gene expression profiling, either it's a transcriptomic RNA sequencing data or microarray data, the results are inconsistent for the distinguishing between these two stem cells. So epigenomic also inconsistent, but whereas combinational methylation or gene expression data, multi-omics integration data, it shows consistent result. So so therefore it can be useful. So that's why people before microarray, people was using microarray for the more information. Then people microarray to move to RNA sequencing. After RNA sequencing, side by side there was methylation sequencing, attack sequencing, microRNA sequencing, epigenomics. So after RNA-seq, people started again moving to single cell RNA sequencing. So therefore, the with technology, like the information also is moving and it can be useful for the organoids or stem cell research. So here the key points for the research or bioinformatics analysis. First, you need like replication and proper design of your study. then your quality control gets metadata. Second is like for pipeline, first some kind of test data or document or version everything. Then the interpret those data in terms of like pathway, biological feasibility. Then aim of the translation, like for mechanism to the prediction and to validation of your experiments. So this is the key point for the bioinformatician for using in stem cell research. So that's all, and suggested is like just pick a data set, then define endpoint, like don't pick some kind of random data, and build reproducible pipeline so that when you do some analysis, that fixed your script and your pipeline so that you can use same pipeline for many other data also, so you don't have to change again and again. Then you give your like quality control, check your quality control. That's all. So people can use that. And all in our all my slides, this blue color link, people can visit and get all the cheat codes and R code, and they can learn freely in all the softwares and other things whatever I have mentioned here. Or people can contact me through this mail ID sunirmal123@korea.kr. Anytime they can mail. And yeah, that's all, and thank you, and thank you for the opportunity. Yeah.
Uh, Thank you, Dr. Sunirmal Sheet. This is wonderful information from your research. So definitely, students and professor can get more idea from your research and more informations. So from audience side, I have some questions. Can I go with that?
Yes, sure. Yeah.
Yeah, yeah. So can RNA sequencing predict any kind of disease previously before?
Yes,
Uh,
for pre-diagnosis purpose, yes. In that case, for example, if we take cancer, so although
okay.
it's a broad disease, but in terms of any single disease, if you take, so you have to either consider the sample level. For example, for cancer, you need to then collect blood sample. You have to
Okay.
compare with the control. Then you have to compare with the already the patient sample blood level. Then there is a chance that okay, you can find some significant marker, and those markers expression, or those markers how to say, yeah, mostly expression. So you can follow for through any kits or anything for simple test. Then you can say that in terms of diabetes or other disease, how they diagnose. Similarly, you can also follow for cancer also.
Okay. Is more advantage, I mean, more advanced one is single cell RNA than compared to bulk RNA, right?
Yes, single cell
Okay.
RNA is the like now latest sequencing data or sequencing techniques. So where we can get, inside blood also the each cell inside blood also white blood cell, then neutrophil, and among that also population like monocyte and dendritic cell, NK cell, T cell. Each cell population we can know, and their movement, their progression, their expression, and inside that their gene level we can find out. That's why single cell sequencing data is the latest one which people are using, or all researchers we are using.
Okay. Okay. Be grateful and great. Thank you. Thank you so
And
much.
yeah, thank you. Thanks once again for Jiva Hospital and Punjab University for organizing this scientific seminar on stem cell research. Thank you.